Something built like a large dog, but carrying its young in a pouch, is moving through the forests of Tasmania at night, a dozen dark stripes running across its lower back, jaws capable of opening to nearly 90 degrees, almost unheard of among mammalian predators. This is Thylacinus cynocephalus, commonly called the Tasmanian tiger, and for nearly 90 years its story has been treated as a closed case, hunted to extinction by European settlers, with the very last individual dying in a Hobart Zoo enclosure in 1936. Three separate lines of genetic research, developed across 3 different decades, have since taken that simple story apart. One revealed the species was already in trouble long before a single European set foot in Australia. Another suggests the animal was still alive for decades after its official extinction date. And the third is currently attempting to reverse that extinction altogether, using the genome of the very animal being blamed for finishing it off. Start with what the thylacine actually was, because the name causes constant confusion. Despite the nickname, it was not a tiger, not a wolf, and not related to either. It was a marsupial, carrying its young in a pouch the same way a kangaroo does, and its closest living relatives today are the Tasmanian devil and the numbat. Its dog-like skull and body shape came from convergent evolution, the same evolutionary pressure independently producing a similar solution in two completely unrelated lineages, despite the thylacine and modern canines last sharing a common ancestor roughly 160 million years ago. That resemblance, however, stopped well short of behavior. A study examining thylacine elbow joints found a structure typical of ambush predators rather than pursuit hunters, closer in function to a cat stalking prey at close range than a wolf running a target down over open ground, meaning the animal that looked so strikingly like a dog from the outside actually hunted nothing like one. The species once ranged across mainland Australia and New Guinea, as well as Tasmania, but disappeared from the mainland roughly 3,000 to 3,600 years ago, likely outcompeted after the dingo arrived, a predator that never reached Tasmania and therefore never threatened the population surviving there. European settlement brought a formal bounty scheme through the government of Tasmania, beginning in the 1830s, paying hunters for dead thylacines blamed for livestock losses, and by the time legal protection finally arrived on July 10, 1936, it came just 59 days before the last known individual, a male nicknamed Benjamin, died in an enclosure at Hobart's Beaumaris Zoo. The species was not formally declared extinct by the International Union for Conservation of Nature until 1982, decades after that final recorded death. If you're finding this interesting, be sure to subscribe to understand more DNA from the past. The first major DNA revelation arrived in December of 2017, when a research team led by Andrew Pask at the University of Melbourne published the first sequenced thylacine genome in the journal Nature, Ecology and Evolution. The team extracted usable genetic material from a pouch, young specimen more than a century old, preserved in ethanol and held at Museums Victoria, and used it to answer two separate questions at once. The first question involved that uncanny resemblance to wolves and foxes. Comparing the thylacine genome directly against modern canids, the team found that the genes and biological pathways responsible for that skull and body shape were not the same genes doing the same job in both lineages. The two animals had arrived at nearly identical anatomy through largely independent genetic routes, evidence described as consistent with neutral evolution, rather than the two species simply inheriting or converging on the same specific mutations. The second finding was the one that actually reshaped the extinction narrative. Using a demographic modeling technique capable of reconstructing population size across deep time from a single genome, the team found that the thylacine's genetic diversity had been declining steadily for an extended period that began well before humans ever arrived on the Australian continent. The species carried the lowest rate of genetic diversity of any marsupial examined in the study, lower even than the Tasmanian devil, an animal currently endangered by a contagious facial cancer partly linked to its own limited genetic variation. The implication was direct. European hunters delivered the final blow, but the population's genetic health had already been quietly deteriorating for thousands of years before a single bounty was ever paid, a slow-motion vulnerability building for millennia underneath a story usually told as a sudden, purely human-caused disaster. That conclusion did not arrive entirely without warning. 5 years earlier, in 2012, a separate team sequencing mitochondrial DNA from 14 individual thylacine specimens had already flagged unusually low genetic variation across the population, a signal visible in a much smaller slice of the genome long before anyone had the tools to reconstruct the animal's complete genetic history. The 2017 study confirmed that early warning was not a fluke produced by a small sample size. It was the leading edge of a pattern running through the animal's entire genome. That finding on its own would have been enough to complicate the standard narrative. It was not, however, the last correction. In 2021, ecologist Barry Brook at the University of Tasmania released a statistical analysis, later formally published in 2023 in the peer-reviewed journal Science of the Total Environment, built on a database of more than 1,200 recorded thylacine sightings collected between 1910 and 2019. Brook's team applied extinction date estimation models originally developed for cases like the dodo, methods designed specifically to account for the fact that a species rarely vanishes in the exact same year its last confirmed individual is documented, since sparse, credible sightings can continue for years or decades afterward in genuinely surviving populations. Running that model against the full sighting record produced a result that startled even the researchers involved. The most probable window for the thylacine's actual extinction fell somewhere between the late 1980s and the early 2000s, roughly half a century after the animal's official 1936 extinction date, and more than 4 decades after the International Union for Conservation of Nature had already formally written the species off. That result does not mean thylacines are alive today, and Brook has been direct about that distinction in his own public comments, stating he believes there is less than a 1% chance any individuals persist in Tasmania right now. What the study actually demonstrates is something narrower and, in its own way, more remarkable. An animal whose species-wide disappearance is treated in textbooks as a single, precisely dated historical event may have instead faded out gradually across an additional half-century nobody had accounted for, surviving in the most remote parts of the island long after the rest of the world had already moved on from its story. Sightings reported throughout that same window, some involving trained wildlife observers rather than casual witnesses, continue to circulate today, feeding ongoing public fascination even though the rigorous statistical case for present-day survival remains, by the lead researcher's own assessment, extremely weak. That fascination has only grown more recently, rather than fading. A televised segment examining the question aired in April of 2024, and the following month, a wildlife biologist published new photographs online claiming to show a living thylacine in the Tasmanian bush, reigniting the exact same debate Brook's statistical model had already addressed from a completely different angle. The third and most active DNA story involves not reconstructing the past, but attempting to reverse it. In 2022, the same University of Melbourne laboratory that produced the original genome, now operating as the Thylacine Integrated Genetic Restoration Research Lab, entered a formal partnership with the De-extinction company Colossal Biosciences. Their approach centers on the fat-tailed donard, a small, ferociously predatory marsupial that is the thylacine's closest living relative, used both as a genetic comparison point and as a future biological surrogate. In October of 2024, the team announced several concrete milestones. Using RNA extracted from a newly examined, exceptionally well-preserved thylacine head more than a century old, researchers were able to study tissue from the tongue, nasal cavity, brain, and eye, opening a path toward understanding what the animal could taste, smell, and see, details no skeleton alone could ever reveal. The same research identified specific genomic regions responsible for that wolf-like skull shape first flagged in the original 2017 study, and then took the unprecedented step of editing those exact regions directly into living Dannard cells, introducing more than 300 distinct genetic changes into a single cell line, described at the time as the most extensively edited animal cell ever produced. Researchers also reported progress in assisted reproductive techniques for the Dannard, intended to eventually allow an edited nucleus to be carried inside one of the species' own eggs. By August of 2025, lead researcher Andrew Pask had joined Colossal full-time as the company's chief biology officer, with the Melbourne lab formally folded into a new Colossal Australia division. Subscribe to not miss the next one. It is worth being direct about how far that work still has to go. Independent researchers in the field, including geneticist Parwinder Kaur, have publicly cautioned that claims of this scale carry real weight only once they clear formal peer review, rather than being announced first through company press releases, and Pask himself has said the more detailed technical findings behind these October announcements were still headed toward publication. Editing a Dannard cell to carry thylacine-like genetic sequences is a meaningfully different accomplishment than producing a living, breeding thylacine population capable of returning to the Tasmanian wilderness, and the distance between those two outcomes remains considerable, regardless of how much genuine progress the underlying science represents. Even Colossal's own stated timeline for a living, healthy thylacine analog has shifted before and could shift again. A reminder that de-extinction announcements tend to move faster in press coverage than in an actual laboratory. Line up all three findings, and the picture of this animal changes substantially from the one most people carry around. It was never simply a healthy, thriving species abruptly wiped out by bounty hunters within a single human lifetime. Its genome shows a population already in slow biological decline for thousands of years before Europeans ever arrived, meaning the hunting that finished the job was compounding a vulnerability that already existed, rather than single-handedly creating one from nothing. Its actual disappearance, according to rigorous statistical modeling of the physical sighting record, likely took place decades later than the date printed in most reference books, with the species persisting in isolated pockets long after the world had already filed its extinction as settled history. And unlike almost every other extinct animal discussed in this kind of story, its genetic material is not simply being read and reinterpreted. It is actively being rebuilt, gene by gene, inside the cells of its closest living relative, in an ongoing effort that has already produced results but has not yet produced an actual living animal. Three separate scientific disciplines, population genomics, statistical ecology, and synthetic biology, arrived at this same animal from three completely different directions, and each one found a reason to reopen a case most people assumed had been closed since 1982. None of this means the thylacine is secretly alive somewhere in the Tasmanian wilderness today, and the researchers who have studied this animal most closely have been the clearest voices saying exactly that. What it does mean is that nearly every fixed point in the standard version of this story, when it died, why it declined, and whether its disappearance is actually permanent, has been directly challenged by DNA evidence gathered only in the last several years. An animal whose fate seemed completely settled in 1982, when it was formally declared extinct, has instead spent the years since becoming one of the most actively contested extinction stories in modern biology. Not because of blurry photographs or hopeful folklore, but because the genetic evidence itself keeps refusing to close the case.